Logo. National Museum of Natural History. Smithsonian. Text. Hot topic. How do paleoanthropologists identify our evolutionary relatives? Featuring Dr. Bernard Wood, University Professor of Human Origins, George Washington University. May 21, 11:30 a.m. to 12:30 p.m. Eastern Time. Logo. National Museum of Natural History. Smithsonian. On the left is a statue of a humanoid figure with ape-like facial features pulling a tuber from the ground. A speaker's video appears in the upper right.
Hi, everyone, and welcome to today's Hot Topic program. How do paleoanthropologists identify our evolutionary relatives with Bernard Wood? My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray haired woman in a gray shirt with a purple scarf wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us for the Hot Topics or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of your Zoom interface. We are in a webinar format. We can't see or hear you. So as you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen, so we can sort through as many questions as possible. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with a presentation by our speaker, Dr. Bernard Wood, and then I'll join him here to take your questions. And during the presentation, I will also write answers to some of your questions, at least any that I can answer, as will another member of our behind-the-scenes team, Dr. Ryan McCrae. So now I'd like to go ahead and introduce our speaker for today. Dr. Bernard Wood is University Professor of Human Origins at the George Washington University. He studies hominin systematics, comparative anatomy, and soft tissue-based phylogeny in primates, with interest in improving species recognition, phylogenetic reconstruction, and distinguishing reliable phylogenetic signals from noise in the fossil record. He has studied many original hominin fossils and authored or co-authored over 250 scientific articles and 20 books. In 2023, he received the Royal Anthropological Institute's President's Lifetime Achievement Award in recognition of his over 50 years as a leading figure in human origins research, his focus on hominin adaptations and evolution, and his mentorship of numerous PhD students and postdoctoral researchers. And now I'd like to invite Bernard to share his presentation with us.
Bernard Wood's video replaces Briana's in the upper right corner. He has short white hair, wears glasses and a blue-collared shirt, and sits in an office with many bookshelves.
Many thanks, Briana. Can you hear me?
Yes, I can hear you great. We can see you great.
Okay, and you can see the slide?
Not yet.
Okay. Okay.
A slide presentation opens. Logos. CASHP, Center for the Advanced Study of Hominid Paleobiology. The George Washington University, Washington, D.C. National Museum of Natural History. Smithsonian. Text. How do paleoanthropologists identify our evolutionary relatives? Bernard Wood, NMNH Hot Topics. May 21, 2026.
How about that?
Perfect.
Okay.
All right, take it away.
Okay. Okay, well, hello, everybody, wherever you are. Thank you for joining us. I'm going to just run through some images that I still call slides, even though they're not in a projector, and I will get going.
Next slide. A side-angle profile picture of Charles Darwin shows him with gray balding hair and a long gray mustache and beard. Text. Charles Darwin, 1809 to 1882. In 1875 by Walter William Ollis by kind favor of Christ's College, Cambridge.
This is a picture of Charles Darwin, and the reason I'm showing you the picture is because of Darwin's contribution, which includes the notion that all living things are part of a single tree of life. So this is an oil painting of him at the college where he was in Cambridge. And the next is
Next slide. A black and white photo of Darwin with the same hairstyle, wearing an overcoat. Text. Herbert Barrow, 1882.
This is an early photograph. It's not long after Henry Fox Talbot invented photography. And it's a photograph of Darwin the year that he died. So this is the metaphor that he introduced.
Next slide. A black and white inked drawing of a tree with multiple tiny branches making a dome top. Text. Tree of Life. TOL.
This is a tree. You can imagine this is three-dimensional and not the two-dimensional tree you see here. You can see it has a trunk, and so there is a common ancestor. And all the species alive today are on the surface of the tree of life, if you can imagine that. And all the animals that have ever been alive in the past, they are within the tree of life. And we are at the end of just one of the twigs which runs towards the surface of the tree. And my task today is to try and explain to you how we identify the adjacent twigs, the ones that are closest to us. The term I've already used is species, and it's part of the way that we describe the world.
Next slide. Text. Some not so everyday examples of taxonomies with species equivalents. Powered vehicle, automobile, Rolls-Royce, Silver Shadow. In bold, 1970 Silver Shadow 2. Back to non-bold font, 1970 Silver Shadow 2 chassis number 1970114.
Now, rather than dwell on the species, let me give you a couple of examples of taxonomies of which a species is a part. It's the taxonomy, how we describe living things and how we describe living things in the past. So let me give you an example of a taxonomy. So we have an inclusive category. Let's say it's a powered vehicle. You know, it could be a spaceship, or it could be a commercial airliner, or it could be an Amazon delivery truck. And then automobiles are a subcategory. Rolls-Royce automobiles are a subcategory of automobiles. The Silver Shadow model of Rolls-Royce is a subcategory of Rolls-Royce. And the model that was begun in 1970 that's called the Silver Shadow 2 would be the equivalent of a species if we were applying it to cars. If you're not a car person, how about saying something like fine arts?
Next slide. Text. Fine arts. Paintings. Oil paintings. 20th century oil paintings. Picasso. In bold, Picasso's anti-war paintings. A black and white Picasso painting with a jumble of people with chaotic expressions and body parts is at the bottom of the slide. Text. Guernica.
Then paintings are a subcategory of fine arts. Oil paintings are a subcategory of paintings. 20th century oil paintings are a subcategory of oil paintings, and Picasso's paintings, and then Picasso's anti-war paintings would be the lowest, the least inclusive, the most exclusive category. And that is one of them, but you can't classify a single painting. You put it into the species equivalent, which is Picasso's anti-war paintings. So now I've confused you with trying to give you metaphors. Let me explain what I'm trying to do in this talk.
Next slide. Text. 2 questions. The first focuses on the present.
So I'm trying to answer two questions. The first is, who are our own species' closest living relatives, and when do we share a recent common ancestor with them?
Next slide. Text. The second question concerns the past.
And the second question, does our twig of the tree of life consist of just the ancestors of modern humans, in other words, our ancestors? Or do we also have close fossil relatives that did not make it onto the surface of the tree of life? So those are the two questions.
Next slide. In the top left is an orange album cover for the Beatles' Yellow Submarine, Eleanor Rigby. It features the four members of the Beatles. Text. 1966. Tomorrow Never Knows, Rain, paperback writer, and Eleanor Rigby.
The first time I became aware of human evolution was when I took a course in human evolution in 1966. It's a long time ago. I worked for 12 years in Liverpool, so I'm expressing that in terms of the Beatles. And 1966 was when the LP came out, The Yellow Submarine, and you can see the songs that are in it. So it's a long time ago.
Next slide. A chart labeled family has a vertical axis on the left that runs from 0 at the top to 11 at the bottom. Across the top there are photos, one of a Time magazine cover with a woman's face, and then 3 pairs of photos of different types of apes. There is a solid red line coming down from the Time magazine cover, ending in a question mark. Below that is the text, hominids. A dotted red line separates the magazine cover and hominids section from the right side of the chart, with all the apes. A dotted green line comes from the first ape photo pair, a dotted purple line from the second, and a dotted black line from the third. There is a question mark in between the green and purple lines, and another below the black line. The entire right side of the chart is labeled Pawn Gids.
When I was taught, the conventional wisdom was that hominids, of which we were the sole living example, were separate from pongids, which included the African apes and the orangutans. And so the distinction between us and our closest living relatives was made at the level of the family.
Next slide. The fine arts list returns, this time with oil paintings in bold.
And if we go back to our analogy, that I think would be something like oil paintings.
Next slide. A chart with a vertical axis labeled millions of years ago runs from 0 at the top to 8 at the bottom. It has vertical bars from left to right in different colors, with the key, red, anatomically modern Homo, blue, pre-modern Homo, turquoise, transitional hominins, brown, megadont and hyper-megadont archaic hominins, orange, archaic hominis. Red is on the top left and is a small bar. Several blue bars of different lengths spread to the right, with a turquoise bar to the lower right of the longest blue bar. Further right there is a low orange bar at about 3 to 2 million years ago, the lowest bar on the graph. To the farthest right are 2 brown bars around the same time as the turquoise bar, higher than the orange bar, and lower than most of the blue bars and the red bar.
And in 1966, there wasn't much known about the fossil record of the animals that were thought to be closely related to us. And I'm going to show you a series of diagrams, and the blue ones, they are early hominins that if you saw them on the metro in DC, you might be slightly disturbed, but I don't think you would go to another carriage at the next stop.
He indicates the turquoise bar.
Homo habilis, you probably might go to the next carriage at the next stop.
He indicates the brown and orange bars.
The next ones, Paranthropus robustus and Paranthropus boisei, that were known in 1966, and Australopithecus africanus, you would very definitely move to the next carriage at the next stop, because these are would have been really not terribly like us, maybe Australopithecus africanus a bit, but the Paranthropus robustus and Paranthropus boisei, they would not look at all like modern humans.
Next slide. The chart with the pictures along the top returns with no dotted lines. It is labeled tribe. Now, a solid red line from the Time magazine, labeled hominins, goes down and turns right at 8, where it connects to a solid green line from the first pair of ape pictures, labeled pannins. It turns blue and goes down to about 10, where it turns right and connects to a solid pink line from the second pair of apes, labeled gorillans. It turns brown and goes down, then turns right at around 12 and joins a solid black line from the third pair of ape pictures.
So now what's the situation? So I told you what the situation was in 1966. Now we separate modern humans from the African apes and orangutans at the level of the tribe. And so we are in the hominini, and so we use the informal term hominins. The chimpanzees and bonobos are in the panini, and so we use the informal term pannins. We have a lot better information, but still not very good information about how they are related to each other. And based on the genetic differences between modern humans and the living African apes, the hypothesis is that we probably shared a common ancestor with chimpanzees and bonobos about 8 million years ago. Now, if this upsets you, just imagine how upsetting it must be for chimpanzees and bonobos and gorillas, because they are in the zoo, and the people that come to the zoo and buy ice creams and look at them, they are more closely related to one of them, to the chimpanzees and the bonobos, than they are to the gorillas. So I think it will be a lot more upsetting for them than it is for us.
Next slide. The fine arts list returns, this time with 20th century oil paintings in bold.
So, a tribe would be, something equivalent to the 20th century oil paintings.
Next slide. The previous chart returns with a yellow box drawn around the Time magazine cover and the red hominins line.
So my research interest is trying to work out what happens between the hypothetical common ancestor around 8 million years ago and modern humans. And that's what I want to talk about today.
Next slide. A map of Africa with 11 sites marked with colored pins. The greatest concentration of sites is in Eastern Africa, indicated by 8 red pins spanning from Tanzania to Ethiopia. An additional blue point notes a site in Chad and 2 green points note sites in South Africa.
What I need to explain is that the evidence that I'm going to be talking about comes mainly from Africa. But Africa is a big place. And the evidence I'm going to be talking about comes from the East African Rift Valley, which is the red balloons on the right. It comes from a series of caves in southern Africa at the bottom of the picture, and there is one site which is not in either of those places. So when we talk about the fossil record, it has to be with the caveat that we are looking at the fossil record which has been recovered from these 2 places. There clearly must be a lot of other evidence, but we don't have access to it. And the other thing I need to explain, and this is Ryan is going to be helping with the questions.
Next slide. 2 large pie charts appear at the top, with 3 rows of multiple small pie charts below. The pie chart at top left shows the relative amounts of complete information provided by the parts of a modern human skeleton, represented by 9 colored slices in the chart. No slice accounts for more than about 20% of the chart. The chart at top right shows the relative amounts of information scientists have gotten from the hominin fossil record. The slice for permanent dentition or teeth accounts for more than 50% of the chart. Text. McCrae and Wood, 2025, Journal of Human Evolution. Text. Ryan McCrae. In the top right there is a smiling photo of Ryan, who has short brown hair, a short brown beard and mustache, and wears a black collared shirt. He stands in front of trees.
On the top left, there is a pie chart which shows the relative numbers of items of information from a complete modern human skeleton. And you can see, the axial skeleton, that will be all the vertebrae and the sacrum and so on. You can see the teeth and so on. If you look at the pie chart on the right, the large pie chart on the right, that is what the hominin fossil record looks like. So we are reconstructing the hominin fossil record on the basis of mainly information about the teeth, with very limited information about the limbs and the axial skeleton. So you need to take that into account when you are trying to understand what I'm trying to explain to you. So the fossil record comes from limited locations geographically, and the interpretations are based on a very odd sort of individual that would be all teeth and hardly any limb bones and axial skeleton.
Next slide. Text. More evidence, or smarter ways of looking at the existing evidence. An arrow from the text points left to further text. Advance our understanding of human evolution.
So how do we reduce our ignorance about human evolution? You find more evidence, or you think of smarter ways to look at the existing evidence. And that's what we have been doing in the past few years.
Next slide. Text. Geometric morphometics. There are 4 images of a molar. The top left image depicts 3 linear measurements in red superimposed on the occlusal, or chewing, surface. The top center image depicts an outline measurement around the perimeter of the tooth in green. The bottom left image depicts a measurement of 5 different sections of the molar divided by cusp, and the far right image depicts the 3D geometric morphometric approach with multiple points creating a 3D shape measurement. Text. Max Planck Institute, Department of Human Evolution. The 21st of May 2016. Capturing Tooth Morphology. 3D Geometric Morphometrics, 3D-GM, enables morphology to be captured in much more detail. Copyright Matt Skinner. A photo in the bottom right shows Matt. He is bald with a closely shaved beard. He wears a white collared shirt and stands outdoors.
For example, Matt Skinner now works in Leipzig at the Max Planck Institute, but he was a graduate student a long time ago. And you can see on the left of the image, you can see an occlusal view of some teeth. And we thought we were very, very scientific when we measured a tooth. We measured its length from the front to back, and we measured its width in 2 places. We thought that was amazingly sophisticated. And then we gathered information about the shape of the occlusal outline of the crown. And then we got even more sophisticated, and we divided the crown up into the major cusps, and we recorded their areas. So we thought that was incredibly sophisticated. Now people use computer tomography. They use 3-dimensional geometric morphometrics to collect a lot more information. And you can see an example on the right. So that's an example of getting, looking at the existing evidence in a sort of smarter way.
Next slide.
So what are the research questions? How many species are there in the human fossil record?
Next slide. A chart titled Splitter's Taxonomy includes the red, blue, turquoise, orange, and brown bars from the previous bar chart, with many more blue and orange, and a few more turquoise and brown, added. There is also a section of green bars, labeled in the key as possible early hominins, in the lower right, spanning around 4.5 to 8 million years ago. Text. Hominin taxa and site samples according to a Splitter's taxonomy, i.e., Specio's taxonomy.
Well, This is what a splitter like me, in other words, someone who would have a tendency to recognize more species rather than fewer species, this is what it looks like in 2026. If you remember how few species there were in the 1966 version, you can see that all the attempts to collect fossils in the intervening period have been successful. These may or may not be species that will survive the test of time, but nonetheless, this is a sort of speciose interpretation of the hominin fossil record. On the left is millions of years, and so each column is a hypothesis about the existence of that species and its first appearance date at the bottom and its last appearance date at the top.
Text. Each column represents a hypothesis about A, the existence of a taxon, and B, the ages of the existing site samples of that taxon.
So why might there be even more species than are in that sort of speciose interpretation of the hominin fossil record?
Next slide. Text. Why might there be even more species? Spatial scope of the hominin fossil record. Rock availability. Hard tissue evidence does not always preserve the SMRS in extant primate species, e.g., guenons, so is likely to underestimate taxic diversity. Hominins are rare, circa 1 to 2% of the fossil mammals at sites. On the left is an image of a scale with the label, more, on the left dish.
Well, one reason is the spatial scope of the hominin fossil record, and I've already talked about that.
Next slide. The map of Africa returns.
We only have information from a relatively limited part of Africa. There may be hominins that were living in other parts of Africa that we don't have access to.
Next slide. The scale slide returns.
The hominins could be living in a part of Africa, but if they were fossilized, but if we don't have access to the strata in which they were fossilized, then we just don't find them. It doesn't mean they're not there. It's just we're not going to have access to them. We should also realize that the hard tissue evidence that forms the basis of the hominin fossil record may or may not be the reason why primates mate with each other and don't mate with other primates. This is called the specific mate recognition system. So, for example, guenons are monkeys. They mate with each other according to the color of the facial hair. And so green-faced guenons mate with green-faced guenons. They don't mate with blue-faced guenons. And the other reason that there may be more species is that hominins are a very rare part of the mammalian fossil record. They make up about 1 or 2% on average of the mammal fossils found at any site. So you might have a site, you might have quite a lot of bovid fossils or quite a lot of pig fossils, but you may not have hominins. It's just because you haven't been looking long enough and you haven't, they may be there. It's just you can't assume they're not there.
Next slide. Why might there be fewer species? The image of the scale now has a label, fewer, on the right dish.
So why might there be fewer species? Well, if you just think about this, the thought experiment.
Next slide. Text. Thought experiment. Species characteristics. Actual versus observed. A graph has space on the x-axis from actual range to actual range, and time on the y-axis from actual FAD to actual LAD. It is filled with a cluster of gray dots, some faint and some dark, some small and some large. In general, the dots get larger to the right and darker to the bottom. Text. Copyright LD, EB, and BW.
I'm a pretty simple guy, so this is not a very sophisticated image. And it took me a long time to make all these gray circles. But just imagine that there is a species and it's gray circles. Okay. The average length of a mammal species in time is somewhere between 1 or 2 million years. So just imagine the vertical axis is a couple of million years in time. Let's imagine this hominin species was spread over, say, a couple of thousand kilometers. That was its range. So this is a hypothetical species. You and I know they all belong to the same species, but they are not identical because that's the nature of a species. In some parts, they're small. In some parts, the grayness is a little more exaggerated, so they're dark gray. In some parts, they're light gray. Believe me, they all belong to the same species, even though they're not identical. And that is a lesson that some of my colleagues really never learn, that a species has its own variation, and that variation is dictated by climate and environment. In some parts of the range, the individuals will be larger than others because there is more food. In other parts of the range, they will be smaller because there is less food. In some parts of the range, they would have larger chewing teeth, and in other parts of the range, their chewing teeth would be smaller. But this is the same species.
Pairs of red dotted lines cross the graph, catching two large faint gray dots in the intersection of the lines, labeled Site A.
So somebody gets some money to go to Africa to look for hominin fossils, and they find a site, and that site maybe samples 300,000 years, and they discover a hominin and they get themselves a paper in Nature. So that's fine.
The bottom horizontal dotted line lowers, making the intersection larger. Another pair of faint large dots is circled below the first, labeled Site B. Text. Same species? Yes.
You and I know that this other site, it's sampling the same species. Because it's the same species, even though the information is really valuable, the researchers try and submit their paper to Nature or Science, but it's rejected, and they end up publishing it in the Journal of Human Evolution.
3 small, dark dots are circled on the far left, not inside the intersection, and labeled Site C.
Then somebody gets some money and they go to a different part of Africa, and they think, wow, we found some stuff that doesn't look like the large light gray circles. The circles are smaller and they're slightly darker. Wow, we can write a paper for Science or Nature saying we found a new species, but you and I know that it's the same species.
Text. Same species? Yes. Next slide. The bar chart returns, now titled Lumper's Taxonomy. It has far fewer, larger bars in each color. Text. Hominin species according to a Lumper's taxonomy, i.e., Les Specios taxonomy. Updated version of Wood and Boyer, 2016.
So that's why one needs to think that some of the more speciose interpretations may not be correct, although I'm inclined to think that they will eventually be shown to be more correct. But nonetheless, if we express the hominin fossil record in the smallest number of species that we can use to interpret it, it still looks like this. And it's still the case that from just more than 4 million years ago until the recent past, until the last Neanderthals or Denisovans died out, there has always been more than one hominin existing in the world.
Next slide. Text. Research questions.
So how are the species related to one another? Now, if there's more than one hominin existing in the world at the same time, they can't both be our ancestors. And so we are, the problem here is to sort ancestors from close relatives.
Next slide. The chart with the photos along the top returns. Text. Does our twig of the TOL, the red line in the yellow box, consists of A. Just ancestors of modern humans, or B. Do we also have non-ancestral close relatives that did not make it onto the surface of the tree?
So the question is, does our twig of the tree of life consist of just ancestors, or do we also have non-ancestral close relatives?
Next slide. A black and white photo shows people in formal attire gathered around a table with a lavish centerpiece. All of the people have a red R labeling them. Text. Martha and John Wells' golden wedding anniversary celebration, held at 109 Church Street, Stoke Newington, North London in 1933.
This is a picture of my father's family, and it was a golden wedding anniversary, and the picture was taken in 1933. All the people in the picture are my relatives.
Yellow A's replace the red R's for everyone except one woman in the front. Ancestors equals 5, close relatives equals 1.
On the left is my father's father. If you go up a bit and look at the A, that's my father. If you go to his left, but our right, that's his mother. If you go down, that's her mother. And if you go to the right, the other side of the rather nice table decoration, that's her father. The R is Aunt Kitty. She was delightful, but she was not my ancestor. She was a close relative. Okay. Aunt Kitty was a close relative and not an ancestor. So in that picture, the ratio of ancestors to close relatives is high in favor of ancestors.
Next slide. A black and white photo shows a family gathered on a lawn, holding 2 toddlers among them. All the adults are labeled with red R's. Text. Bernard Wood's christening party held at Spring Hill, Hackney, North London in 1945.
This is my christening, and you can see that there's a mixture of relatives and family friends and godparents, and the relatives are identified there. You can see it. Well, you don't have to be a genius to realize who I am there.
4 people's R's changed to A's.
However, in this picture, the ratio of ancestors to close relatives is actually there are more close relatives than there are ancestors. The only ancestors of mine in that picture are my father's father on the left. My mother is the next A. My father was still away in the war at the time. And then my father's mother is the next A, and then to the right was her mother. So there are 4 ancestors, but there are a larger number of close relatives.
Text. Ancestors equals 4. Close relatives equals 6.
So is the hominin fossil record more like the Golden Wedding or more like my christening?
Next slide. A green check mark appears over the christening photo on the top left and a red X appears over the wedding photo on the top right. Text. Yes, we have non-ancestral close relatives as well as ancestors, so more like a more extreme version of the ratio at BW's christening than at the Golden Wedding celebration, i.e., 1 to greater than 2 rather than 5 to 1.
It's probably more like my christening. In other words, I think there are a whole bunch of close relatives in the hominin fossil record that aren't necessarily our ancestors.
Next slide. The splitter's taxonomy returns.
So just to remind you of the rather specious interpretation of the hominin fossil record, most of those will be close relatives and not ancestors.
Next slide. The lumper's taxonomy returns.
And even in the lumpers interpretation, the balance would still be most of those will be close relatives and not ancestors.
Next slide. Text. In theory, reconstructing relationships should be simple. Shared morphology means shared evolutionary history. So the more morphology species share, the more closely related they should be, but
so in theory, reconstructing relationships should be simple, because the more morphology you share, the more closely related you are. But there is a bit of a sort of but I wish life was that simple, but it's not.
Next slide. A painting shows men in black overcoats grouped around a man wearing a beige overcoat, sitting at table with several humanoid skulls and papers spread out on it. The man in beige measures one skull. Text. The Piltdown Committee by John Cook, 1915.
And this is a picture of the Piltdown Committee looking at the Piltdown discovery. And the guy who you can hardly make out on the right is the person who I'm going to focus on.
Next slide. A black and white photo on the left shows a man with slicked back dark hair wearing a dark suit and tie. Text. Quote, when identical or nearly similar forces or environments act on two or more parts of an organism, the resulting correspondence is called forth, and the several parts in the two organisms will be nearly or exactly alike. I propose to call this kind of agreement homoplasis or homoplasy. Lancaster, 1870, page 39.
And he was a guy called Sir Ray Lancaster. He was the director of the Natural History Museum in London. Apparently, he was a perfectly awful manager, but he was interested in relationships, and he wrote in 1870, when identical or nearly similar forces or environments act on two or more parts of an organism, the resulting correspondence is called forth in the several parts of the two organisms. This is very flowery language, it's very Victorian, will be nearly or exactly alike. So he's really saying that you can get very similar sorts of animals because they are responding to the environment, not because they are necessarily closely related. And he says, I propose to call this kind of agreement homoplasis or homoplasy. Neither of them really caught on as an idea. Eventually, his idea was called homoplasy. And so homoplasy refers to features shared by two taxa that were not present in their most recent common ancestor. Such morphology gives the false impression that the two taxa are more closely related than they really are, and it complicates attempts to reconstruct phylogenetic relationships. And we know that's the case. We know that homoplasy is rife in groups like cetaceans, which have a much, much better fossil record than hominins. And so it would be crazy to imagine that hominins were not affected by homoplasy, which means that trying to recover relationships is a rather harder problem than we would like it to be.
Next slide. The chart with the photos across the top returns, with the text. How you tell the ancestors of modern humans from our non-ancestral close relatives remains a significant challenge.
And so it really remains a significant challenge.
Next slide. The splitter's taxonomy graph returns, with a yellow arrow pointing at the largest green bar, running from around 6.5 to 5.5 million years ago, labeled AR.Kadaba.
So if we go back to this sort of specios interpretation, and we look at the green ones down in the bottom right, these are all taxa that their discoverers were convinced were the earliest hominins. Now, they can't all be the earliest hominins. And so what are the options for creatures that don't look like modern humans? They don't look like chimpanzees or bonobos. They're more chimp-like than modern human-like, but they have some things that chimps don't have. they have some things that suggest that they had a slightly different locomotion than modern chimps, and that their diet was probably less reliant on fruit and so on. And so where might these creatures be accommodated in that branching diagram I showed you earlier?
Next slide. The chart with the photos along the top returns. This time the yellow box around the red line under the Time magazine photo is gone. 4 branches now come off of the red line spaced about 1 million years apart each. They all turn upwards and then stop 1 to 2 million years after the branch.
Well, they could be our close relatives. Okay, they could be our close relatives, or they could be on the main red line to modern humans. In other words, they could be our ancestors. But how you tell ancestors from close relatives is a struggle.
Similar branches appear on the green line.
They could be closely related to chimpanzees and bonobos.
A dark gray line appears coming from between the red and green lines where they join at 8 million years ago. It goes to about 4 million years ago with one small branch.
Or they could belong to a group for which there is no living representative, which is actually closer to modern humans than modern humans are to panins and bonobos.
Next slide. Text. Consumer warning number 1.
So, having confused you and tried to persuade you that what on the surface might look a simple problem is not that simple, just a few consumer warnings. Where the fossil evidence is found is not necessarily where the action was.
Next slide. The map of Africa returns.
In other words, a lot of interesting evolutionary history, hominin evolutionary history, may have been going on in places where we have no fossil sites.
Next slide. Consumer warning number 2. Fads and lads in hominin evolution are complex phenomena that are not conducive to simple explanations.
Those first appearance dates and last appearance dates, they're really complex phenomena that are not really conducive to simple explanations. So, you want to avoid, saying, well, the climate changed, so therefore these things evolved. It's probably rather more complicated than that.
Next slide. Consumer warning number 3.
And another consumer warning is that events are what processes look like when they are sampled discontinuously. And so it's easy to see, oh, something happened, but you just don't have access to what happened before.
Next slide. Consumer warning number 4. In the bottom left, there is a black and white photo of a man with white hair and a gray suit, sitting at a tilted desk with his elbow on it and his chin on his hand, giving a wry smile. Text. Albert St. Georgi.
And I'm a great fan of Albert St. Georgi, who said simple solutions to complex biological problems are almost certainly wrong.
Next slide. Text. Indebtedness. Logos. CASHP, Center for the Advanced Study of Hominid Paleobiology. The George Washington University, Washington, D.C. National Museum of Natural History. Smithsonian.
So before I finish, I just wanted to explain how indebted I am to the people who find the fossils, because without the fossil evidence, I wouldn't have had a career.
Next slide. A photo shows 5 men in shorts and button-down or polo shirts standing alongside a blue truck in an arid landscape. The truck bed is low. Text. The hominid, N, gang, Kamoya Kimyu, L, and some of the Kamba workers at the National Museums of Kenya who found most of the hominin and other fossils.
And these are the Kenyans that found most of the fossils from Kubifora that was on the eastern side of Lake Turkana where I worked.
Next slide. Text. Richard Erskine Frere Leakey 1944 to present. A collage of both color and black and white photos shows Richard at various ages, on dig sites, holding skulls, and holding up a National Geographic magazine.
And I want to express my thanks to Richard Leakey, who who was told by his mother in 1968 that he should take this young medical student with him to northern Kenya, and depending on Richard, how frank Richard was being, he sometimes thought that was a good thing, and sometimes thought that maybe, especially when my interpretation of the hominin fossil record differed from his.
Next slide. Text. Thank you.
And I want to thank you all for listening.
Wonderful, thank you. I particularly enjoyed that picture of some of the hominin fossil hunters with the Land Rover that looked like it was sort of cut off halfway up. That was great. Wonderful. So we have a couple of questions. I'm going to actually start with some behind the scenes questions.
The speaker's videos replace the presentation slides.
So I'm gonna ask the first one, which is, considering the numerous ways that scientists can identify and separate species, so these are often called species concepts, are there any that are particularly useful or not useful, do you think, for the hominin fossil record? Or is trying to, follow any number of species concepts ultimately not useful at all?
Well, I think you have to have in mind the species concept that you think intellectually and makes most sense, but then you have to think that the hominin fossil record is made up of hard tissue evidence. And if you think of the guanan example, the facial color of the guanan would not, I mean, there is no fossil record of that. And the problem is that if you look at skeletons of guanans, they are almost indistinguishable. So you would never, if you just looked at their skeletons, you would say there was one species of guanan. But if you go out into the wild, or if you go to a zoo that specializes in guanans, you will realize that that would vastly underestimate the number of species. So the soft tissues don't preserve, and the soft tissues are probably where most of the species of the signals that animals use to decide who to mate with. It's mainly soft tissues.
Yeah, that's a good point. Thank you. So speaking of things that do preserve, talking about genetic evidence, here's another behind the scenes question. Considering sometimes genetic evidence preserves when we're lucky. So considering the genetic evidence of later hominins like Neanderthals and Denisovans interbreeding, and the paper that just came out recently that might include Homo erectus interbreeding with Denisovans, how does this, or does it complicate our picture of how species are separated? So that's the first part of the question. Then I'll ask a second part after.
Well, if you think of living baboons, baboons in Africa extend all the way from North Africa down to Southern Africa. If you look at the baboons in Northern Africa, they're obviously distinct from the baboons in Southern Africa. But if you look at the baboons in Ethiopia, which are in another species, they interbreed with the adjacent species. Now, the interbreeding is not so much that it blurs the distinction. It just means that at the boundaries, you will find individuals that have some characteristics belonging to one species and some characteristics belonging to another, but it doesn't mean that the baboon species aren't a reality. So even if hominins did interbreed, which it would be crazy to think that they didn't, the question, my response will be that the extent of interbreeding did not blur the distinction that we see in the hard tissues in the fossil record.
That makes sense. And actually, you may have already answered the second part of this question, maybe, but so given this interbreeding, which I think we would expect, do you think that this interbreeding was present all throughout our evolutionary tree, or is it really a more modern hominin phenomenon with these like very closely related taxa like humans, Neanderthals, Denisovans?
My sense is that it would have been, there would be more of it recently.
Mm-hmm.
You should understand that, as soon as our ancestors looked at all like modern humans, I lose interest. And so, it's wonderful to read these papers about sort of Denisovans and Neanderthals. But I'm sort of more interested in creatures that really look sort of different.
Mm-hmm.
So different, if you take Paranthropus boisei, it's such a weird creature.
Mm-hmm.
There's no living primate that makes a reasonable model for trying to understand how this creature made it, how it lived. And so I'm sort of more interested in the relatives that come to your Thanksgiving dinner that, you're not quite sure about, but somebody tells you that you're related to them, so they should come to the Thanksgiving dinner. So I'm more interested in those, or their
Um,
equivalent.
so actually, this leads on to another behind the scenes question. Maybe you just answered it. Do you have a favorite species of hominin or a favorite fossil? And if so, why is that your favorite?
Um, well, my favorite fossil is the type specimen of Paranthropus boisei, and it's called Alderby hominid 5. And when I was doing my research, it was actually in a museum in Dar es Salaam. And Leslie Aiello and I went to see the material there. We clearly didn't have to make an appointment because we think that nobody had been there for 5 years before we went there. The safe in which the skull was in a cupboard. There was no light bulb in the room, and there was no light bulb in the cupboard. So we had to go out to a store and buy some light bulbs. And so we eventually got OH5 out of the safe. It is the most fantastic specimen you will ever see in your life. It's just amazing because it's so perfectly preserved. Much of it's perfectly preserved, but it's so weird.
Hmm.
So the answer to that would be OH5 on Paranthropus boisei.
All right, thank you. That's good to know. And if I'm going to get you a thank you gift for doing this talk, it might have an OH5 theme. So I'm going to ask a question that was sort of going off one of our audience questions. The initial question was about why are Denisovans not on your initial species list? And the answer was, well, they hadn't been found yet
Yes.
in that 1966 list. But to sort of go off of that, are there any potential species of hominins that you don't include on your chart that you think might be species, but like are maybe less well accepted?
No, I think most, the chart includes all the runners and riders, I think.
Okay.
Even the ones that, aren't going to, they aren't going to be placed in the race, but nonetheless, you
Okay.
should have them on the board.
Got it. Um, so I might ask you a few sort of career related questions. One is, so I know you started your career in medicine, you were dean of a medical school. How did you get into paleoanthropology?
Well, I got into paleoanthropology because when I was a medical student, I did an extra degree in anatomy, and I did a course on human evolution. And the course on human evolution was taught by a guy called Michael Day, and he had worked on the fossils discovered by Lewis and Mary Leakey in Alderby Gorge. Um, Michael Day's wife, you did ask me. Michael Day's wife was a primary care physician. She used to look after Mary Leakey, and they were very good friends. And so, you know, somehow, sort of Mary said, well, I think this, young man who's, a student should go up to northern Kenya. I'll have a word with Richard. And so basically she went home and said, Richard, I think you should invite this guy. So I went, I, I enjoyed it. Richard's tolerated me. I mean, you've been in the field, you know perfectly well, that the people you want to be in the field with are not people who drive you crazy,
Yes.
And so I had enough, I had enough sort of wisdom to make sure that I wasn't a nuisance, and we got on well together, and he invited me back, and he invited me to describe the fossils that he was finding along with other people. And so I did surgery. And I had to go back to do the exams for the fellowship of the Royal College of Surgeons. And I kept putting off doing that exam because I kept doing more research. And eventually I was on the, eventually I became an examiner for the anatomy exam for the Royal College of Surgeons. And I thought, I'm not going to, now I'm an examiner, I just don't have the chutzpah to take the exam, because supposing I failed it. And so basically, so I just sort of left medicine. I mean, I worked as a surgeon for a decade on and off, I did surgery part-time.
Okay.
But then I eventually stopped.
Okay. I'm going to turn to an audience question. And so Walter asks, how much detail of the surrounding past geological environment is studied and correlated with the description of a species? Could this contribute to homoplasy?
Yes. Yeah. I mean, I think it's what sort of drives homoplasy, which is why we are so keen to learn from our colleagues who are expert in interpreting the sediments, interpreting the other fossils, to try and paint a picture of the habitat that the early hominins were living in. So I think that's a very astute question.
Okay. Let me, I'm just looking at some more behind the scenes questions. So can you, to follow up on this one, can you elaborate on homoplasy? Are there particularly important examples in the fossil record, or good illustrative examples?
The problem is that in the fossil record, you don't have, the reason that we know homoplasy exists is because people use molecules and latterly the genetic information about DNA for working out how animals are related.
Mm-hmm.
So we have information about relationships which is independent of morphology.
Yeah.
The problem in the fossil record is that we have that, for Denisovans and for Neanderthals, but there is really no sort of good DNA information much older than about 500,000 years. And so the problem is we don't have a non-fossil evidence related line of evidence about relationships.
Yep.
And so we just have to do our best. But I think, you need to look around you and see, well, is there homoplasy? Yes, there is. And why do you think hominins should be somehow exempt from homoplasy?
Right.
And I just think it's just crazy to make.
Yeah. I'm going to turn to another audience question. So Stephen asks, will the new science of protein analysis, as opposed to DNA, help in distinguishing species, do you think?
Yes, it is. And it's sort of almost counter, intuitive. You would have thought that sort of proteins would be easier to get at than DNA, but it turns out that the proteins. So, for example, there is a difference in one of the proteins in our teeth. It's a protein called amelogenin. And there is a sex difference. So that helps us identify fossils as males and females. So for example, Paranthropus boisei, one of the things that interests me about it is that the males are not just larger versions of the females, there are shape differences between the males and the females. And so if we have non-morphology related ways of telling males and females, it allows us to say that is what we call within species or intra-specific variation. It's not the difference between two species. Now, you know, there could be a male of one species and a female of another species, but if your working hypothesis is that this is one species, and I think these are the large males and these are the small females in the species, and that is confirmed by the amelogenin, then that, you know, makes you feel good.
Exactly. All right, I'm going to go to another audience question. Tad says, great talk. I love the family photos as a teaching tool. So in this talk and others, I hear a lot about our common ancestors with other living primates, and Dr. Wood identifies that at about 8 million years. But has anyone tried to trace our ancestry back earlier than that, like all the way back to when the asteroid hit 65 million years ago and mammals first began to emerge?
Yes, but it's a different scale of problem. I mean,
Yeah.
you know, if you're in research, I mean, you know, it's like going on safari. You know, you wake up in the safari lodge and you take your coffee outside and you see elephants and giraffe and you see baboons and you see everything. You know, if you're a researcher, you need to choose which of these animals you're going to focus on. You know, you can't look at elephants for 10 minutes and then start to do some research on these. And so basically, each of us really has to decide. You know, I have some colleagues who are interested in the origin of mammals. I have colleagues who are interested in the origin of primates. I have colleagues who are interested in the origin of apes. But that's not my bag, you know.
Yeah.
And so I think you have to understand that if you do research, you have to pick where you think you can reduce ignorance. And it's really difficult to do that across, you know, a whole range of research questions. I'm
Yeah,
not sure
that makes.
whether that answers the question.
I think so. Yeah. I mean, I think the answer is yes, and that there are people who ask these questions sort of particular places in a sense on
Yes.
our big
Yes.
family tree. Yeah.
Yes. I mean, you know, if you look at
Yeah.
the tree of life, you
Yeah.
know, there are researchers interested in, you know, in all sorts of, you know, in all sorts of places on that tree. And you have to
Yeah.
realize, you know, if that was literally the tree of life, the vast majority of that tree would be bacteria and would be,
Yeah.
you know, would be organisms without backbones and things like that. And so I'm only interested in a tiny part of that tree.
Yeah, exactly. All right, another audience question. John asks, and this may be a bit outside of the fossil record, but lacking evidence of gene flow due to the age of samples, is there any degree to which we can see flow or sharing of lithic or other technologies to indicate relationships between populations? He says, or are the sample sizes too small?
That's a very interesting question and a very sensible question, because lithic technology is lasting evidence of behavior. And you know, you could say, well, you know, this hominin made stone tools this way, this hominin made stone tools that way, and if you could link those. The problem is that, you know, there are only a few ways that a stone can fracture.
Yeah.
So basically, you know, you have the homoplasy problem coming in, that, you know, if you bash one stone against another, then, you know, just a limited number of things could happen. And we have been working, you know, and in fact, we've written about this pretty recently, and it's a struggle to link stone tools with hominins.
Yeah.
And you have to realize that hominins were probably doing a hell of a lot else other than making stone tools. It's just that we tend to, you know, because that is lasting evidence of behavior, that's what
Mhm.
we focus on because it's there.
Exactly.
But we need to remember that, there may be lots of behaviors that they were indulging in that leave no lasting evidence.
And I agree that sort of assuming that a stone tool technology was only made by one hominin species is, I think, an assumption we should question. So,
Yes,
yeah,
yes. It's certainly not where I would sort of start, I think.
yeah. Here's another audience question from Misha who asks, perhaps this is an oversimplification based on teaching children, but when I was in grammar school, we were taught that species are species because they do not breed with members who are not of their species.
That's right.
Given that modern humans, as I understand, have both Neanderthal and Denisovan DNA, how useful is such a fine distinction as species?
Well, I think it's still useful.
Mm-hmm.
I mean, the fact that it may not be the definition that we were brought up with in a biology textbook, it still means, that let me put it this way, if it wasn't useful, then everything would blur together.
Yeah, yeah.
And it doesn't,
Mm-hmm.
it doesn't.
Mm-hmm.
Most of the hominin fossil record, Paranthropus boisei is very distinctive. It's really difficult to confuse it with early Homo.
Mm-hmm.
However, there are some specimens, that have a bit of this and a bit of that, but
Mm-hmm.
mostly it's very distinctive.
Yeah.
And so I think interbreeding, I think is a reality, but it doesn't blur the major distinction that is preserved.
Yeah. That makes sense to me. All right, I think we have time for one or two more questions, and I'm going to privilege a behind the scenes question. So this question is the number of species, the number of fossils, the general understanding of human evolution has increased through time and for sure over the course of your career. Do you see this increase being linear, exponential? Is it tapering off? How would you predict that pattern to continue into the future, especially with new techniques like ancient DNA and proteomics?
That's an interesting question. I mean, I think there must come a point when we are not going to collect any more information, which is going to change, an interpretation very much.
Mm-hmm.
My experience is that when you only had a few species, everything looked pretty obvious.
Mm-hmm.
There was a time when, you had rather more information and things look, rather confusing.
Mm-hmm.
And you collect a little more information and some of that, some of the confusion gets answered.
Mm-hmm.
My sense is that you have to remember what your research questions are,
Mm-hmm.
not get sort of deflected by the latest sort of shiny object. And
Yeah.
and so that's my reflection from doing this rather longer than probably anybody with any sense should have done it.
Well, I think we've enjoyed the perspective from the course of your career, and I will ask the last question. Also, this question is actually from me. So, kind of following on the question we just asked, what is the discovery over the course of your career that was most surprising to you?
He laces his fingers and squints in thought.
It's surprising. Well, my discovery or other people's?
Anybody's discovery,
Oh,
or
um,
your discovery. I say
no, no,
interpret it
no,
as you wish.
no, no. Um, you know, I'm not sure there was anything that was most surprising. Sort of something like Sahelanthropus.
Mm-hmm.
I wouldn't have necessarily um, predicted all the details, but you sort of in your head
Mm-hmm.
knew that there were creatures likely to be creatures like that.
Mm-hmm.
I'm sorry, I'm not. I guess I'm not being very. There's nothing sort of terribly surprising. And to be honest, if you're a half decent scientist, you shouldn't be surprised.
Well, I was going to say, I think the fact that that nothing springs to mind just means that you've been thinking really broadly about this, and that there hasn't been anything so left field that it
Sure,
didn't really fit in our understanding in general.
sure.
Yeah.
There are a few things, where people say, "Oh, well, you know, Sahelanthropus isn't a hominin, it's a fossil gorilla," and you sort of, you think, well, what were you drinking? It's not a fossil gorilla because it doesn't look anything like gorillas. It just,
Hmm.
if you looked at it from the other side of the room, it might look like a gorilla, but if you look at a car at the other side of a large car park, it's going to have an engine compartment, a passenger compartment, and a trunk. Well,
Hmm.
that's, most cars look like that unless they're SUVs, and that's not terribly helpful. So, yeah, yeah.
Yeah, that's great. Thank you. All right, so I will wrap up. So we'll conclude today's virtual program. Please join me in thanking Bernard for sharing his work and his thoughts with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae, who works with us in the Human Origins Program, to our donors, volunteers, and viewers like you, and finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. This was our last hot topic of the spring season. We'll be back in the fall starting on September 24th, so keep an eye on your email for updates about the fall season of Hot Topics. We've put a link in the Q&A where you can find information about our other upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed about upcoming programs, Hot Topics, and others, and to learn more about the museum's research and exhibits. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you to Bernard, thank you to the audience, and we will see you in the fall.
Logo. National Museum of Natural History. Smithsonian.